So now let's take a look at the embryonic stage and talk about the rest of fetal development. So the embryonic stage is going to begin about 16 days after conception, after fertilization has occurred, and this is when the germ layers are present. So those 3 primary germ layers that I mentioned in the previous video, this is when they're now present. And over the next 6 weeks of development, this is when the placenta will begin to form in that uterine wall. And this will become the primary means of nutrition for that developing embryo and later on for the fetus as it gets larger. And we know that during this time, those primary germ layers will also differentiate into all of the organs and organ systems of that fetus. So all of the organs should be relatively present and relatively developed by about 8 weeks of gestation, and this is when we transfer from the embryonic stage into what's called the fetal stage. So as that conceptus starts to embed in that endometrium, we talked about how the trophoblasts will start to digest certain endometrial cells, and then they'll transfer those nutrients to that inner cell mass. So this is how that inner cell mass will get its nutrition for about the first week of development after implantation. After about 11 days, at that point the placenta will begin to form, and eventually it will take over the role of providing nutrition to that developing embryo and fetus. And so this will happen by about week 9. This will be the dominant provider of nutrition for the fetus by around week 9, and by week 12 it will be the only mode of nutrition for that developing fetus. So let's talk a little bit about the placenta. We know that this is a disc-shaped organ. It's going to attach to the uterus, typically on one side. Many times it attaches on that posterior body wall, but sometimes you may have what's called an anterior placenta, which can make it a little more difficult for the mother to get good images of the fetus as it's developing, because the placenta can sometimes get in the way during that process. But we know that the fetus will be attached to the placenta through what's called an umbilical cord. And that uterine surface of the placenta is going to be kind of shaggy because it has these chorionic villi, these little extensions that embed in that endometrium, whereas the fetal side of the placenta is going to look nice and smooth because it's missing those furry or shaggy looking chorionic villi. So the placenta will develop thanks to rootlets of that trophoblast that we mentioned in the last video, penetrating into the lining of the endometrium, and those rootlets will end up becoming these chorionic villi. These are going to be invading the uterine blood vessels and kind of being surrounded by pools of blood from the mother's body, and then they will eventually merge to form a single blood-filled sinus, which will again consist of blood from the female's body, which can provide nourishment to that developing fetus or embryo. Now the embryonic mesoderm will grow into each villi, or villus, and then form blood vessels that connect the embryo by the umbilical cord. So the embryo will be physically connected to this umbilical cord and to this placenta through 2 umbilical arteries and 1 umbilical vein. So the 2 umbilical arteries are going to bring blood from the fetus to the placenta, so this will be waste-filled blood, consisting of nitrogenous waste, carbon dioxide, things like that. And the umbilical vein is going to return blood from the placenta to the fetus. So this blood will be oxygen-rich, nutrient-rich, blood that is picking up all the oxygen and nutrients from the mother's body and carrying them to the fetus to supply nourishment to that fetus. So the chorionic villi are going to be filled with fetal blood and then surrounded by maternal blood. And there is a membrane that separates the two. So these 2 collections of blood will never mix with one another. They will always have a divider or a membrane separating those 2 collections of blood. So here you can see an image of the placenta and umbilical cord, which again consists of 3 blood vessels carrying blood from the placenta to and from the fetus. So the boundary that separates the blood between the mother and the fetus is extremely thin. It's only about 3 to 4 micrometers thick, which is very, very, very thin. And that makes it very permeable to substances, things like nutrients, which needs to pass from the mother to the fetus, and the oxygen, glucose, things that the fetus needs to survive, Also allows fetal waste to pass from the fetus to the mother's body so that she can eliminate them through her urinary tract or other organ systems of the body. But this can also make the fetus vulnerable because since this membrane is so thin, it's also going to be very permeable to toxins, to medications or drugs, things like nicotine, alcohol, as well as pathogens. Things like viruses and parasites can potentially pass that membrane and make their way into the embryo and could potentially be harmful to that embryo. So here you can see the fetus developing here with all of the accessory organs still intact. So let's kind of take a look at some of these accessory organs. So there are 4 membranous sacs that are going to be found in or near, or around or near the developing embryo. So these are the chorion, which you can see in this kind of pink color here. It's going to surround the fetus as it's developing. It's also going to be surrounding the amnion, which you can see in blue. The amnion is going to enclose the fetus, and it's going to begin to fill with a fluid called amniotic fluid as that fetus is developing. That fluid is going to be there to cushion that embryo. It's also going to be there to protect the embryo from trauma, to protect it from infection, to help prevent drastic temperature changes around that developing fetus. It gives the fetus some space to be able to move around. It prevents the body parts from kind of adhering to one another during development, so it gives them some room to move around. It's going to enable that embryo to develop symmetrically and prevent it from having any kind of odd congenital deformities for the most part during development. It's also going to stimulate the fetus to learn to breathe because they're going to be breathing in this amniotic fluid as a way to practice breathing techniques. The fetus will also be swallowing this fluid during development and urinating it back into that amniotic sac. And then when the baby is ready to be born, this amniotic sac will have to be ruptured. That's when the water breaks, and that amniotic fluid will leave the female's body before the childbirth process will actually begin. Now again, the chorion you can see in this light pink surrounding the amnion. It's going to play some nutritional and hormonal roles in the pregnancy. It's going to help form part of the placenta, and it's going to help enclose the rest of the conceptus, including this amnion and the fetus. Next we have this umbilical vesicle, which is also sometimes known as the yolk sac. So this is, in animals that have shelled eggs, like reptiles and birds, the purpose of this is to enclose the egg yolk, which would be providing nutrients to that developing embryo. But in mammals, including humans, it contributes to the formation of the digestive tract later on during development. It produces the first blood cells for that fetus, and it's going to produce stem cells for future egg or sperm cells to be able to develop from later on. Now the allantois, you can see here, kind of in the middle, this will begin as an outgrowth of the gut, and in mammals, it's going to form the foundation of the umbilical cord, which will help attach that fetus to the placenta later on in development and provide nutrients to that fetus and allow the fetus to be able to remove waste from its body. This will also become part of the urinary bladder in that developing fetus. Now let's take a look at the placenta here a little bit more closely. You can see the myometrium and endometrium of the uterine wall. And then here you can see that pool of maternal blood, which is surrounding the fetal blood, this chorionic villi, which are emerging from or part of the placenta. So these chorionic villi contain fetal blood, and you can see that blood is being transmitted to the fetus through the umbilical cord, through this one large umbilical vein and 2 smaller umbilical arteries. Remember, the arteries are carrying blood away from the fetus, whereas the umbilical vein is carrying blood towards the fetus. So you can see the vein is colored in red because it's carrying that oxygen-rich blood, whereas the 2 umbilical arteries are colored blue because they're carrying more oxygen-poor blood back to this chorionic villi. And so this is where the gas and nutrient exchange is occurring, is across this placental barrier. It's very thin membrane that is separating the fetal blood from the maternal blood in the placenta. Now let's move along to embryonic folding and talk a little bit about this. So at about week 4, the embryonic disc will go from flat to cylindrical. And at this point, the embryo will grow and it will fold around this umbilical vesicle and end up becoming more C-shaped. And so we know the lateral margins of the disc fold to form the ventral surface of the embryo and enclose the primitive gut, which is going to be the future digestive tract of these embryos. So here you can kind of see this primitive gut here, which is forming through this embryonic folding process. We also know a body cavity will form within the mesoderm and become divided by a diaphragm. At this point, this will give rise to the thoracic and peritoneal cavity of the developing fetus. You can see that peritoneal cavity forming right here below. We also know that the development of distinct organs from the embryonic germ layers is called organogenesis. So this is where those organs will develop from these layers of the embryo here. We know that the neural tube will start to form, and that neural tube will end up developing into the brain and spinal cord later on. The mesoderm will begin to fully develop at this point as well, and get segmented into blocks called somites, which will give rise to the vertebrae of the fetus, the ribs, the skeletal muscles, and the dermis of their skin. So by about week 9, all of the organ systems of the fetus are essentially going to be present. That fetus will be about 3 centimeters long, and this is where some of the bones will start to harden and calcify, skeletal muscles will start to form and start to provide contractions. The heart will start to beat. You can typically pick up that heartbeat in an ultrasound by usually even week 6, but definitely by week 9 you'll see a nice strong heartbeat. And we know that the head is going to be about half the total body length by about week 9 as well. So in organogenesis, again, this is where those 3 layers, those 3 primary germ layers are going to be developing into organs of the fetus. The ectoderm we know is going to give rise to the epidermis of the skin, hair follicles, erector pili muscles, and cutaneous glands of the skin. It's going to give rise to the nervous system, the adrenal medulla, the pineal and pituitary glands. It gives rise to the lens, cornea, and intrinsic eye muscles. It's also going to give rise to internal and external ear structures, salivary glands, the epithelia of the nasal cavity, oral cavity, and anal canal. The mesoderm will give rise to bones, bone marrow, and cartilage. It's going to give rise to skeletal, cardiac, and most smooth muscle. It's also going to give rise to the adrenal cortex, the middle ear, the dermis, blood and lymphatic vessels, bone marrow, lymphoid tissue, the epithelium of the kidneys, the ureters, the gonads, and genital ducts, and the mesothelium of the abdominal and thoracic cavities. The endoderm, which you can see in yellow in this diagram here, is going to give rise to most of the mucosal epithelium of our digestive and respiratory tracts, the mucosal epithelium of the urinary bladder and parts of the urethra, epithelial components of the accessory reproductive and digestive glands, the thyroid and parathyroid glands, and the thymus. So you don't have to worry too much about the specifics of those organs that develop, but I just wanted to go through them briefly with you, the organs that are coming from those 3 primary germ layers. So here you can kind of see those stages of prenatal development. This is an 8-week embryo versus a 20-week fetus with the accessory organs, the fully intact placenta. So now let's look at fetal circulation and talk about some differences between it and normal circulation. So the circulatory system is going to change very drastically from the prenatal stage to the newborn stage. And some changes between these are going to occur because of the difference in how that fetus needs to get its oxygen and nutrients versus how a newborn baby would need to. So we know that the fetus is going to be getting its nutrients through the umbilical cord, right? So through those umbilical arteries and the umbilical vein, that is how the fetus will be exchanging gases and nutrients from its body to the mother's body and vice versa. And so it's going to have some different aspects to its fetal circulation compared to an adult's circulatory system. So we know that blood comes to the fetus through the umbilical vein, and that oxygen and nutrient-rich blood then needs to get into the fetus's body. And so since all of the oxygen is present, there will be shunts that will bypass the lungs, and there will be a shunt that will bypass the liver as well. So some of the blood that comes into the fetus's body will go to the liver to nourish it and keep it alive, but most of it will bypass the liver through the ductus venosus, because all of that blood should have already been filtered and cleaned and made pure by the mother's body. And so the liver will not need to play a big role in that process in the fetus's body. And so it can just be bypassed through that ductus venosus. And it basically enters into that inferior vena cava, which is where a mixing of placental blood with venous blood from the body will take place, and it can then flow into the right atrium of the heart. Now, once that blood enters into the right atrium of the heart, most of it, or much of it, will be shunted from the right atrium straight over into the left atrium of the heart through this foramen ovale here. And the reason for that is because this blood does not need to be sent to the lungs because it already is oxygenated. And so we can just bypass the lungs by traveling through this foramen ovale into the left atrium, allow it to travel down to the left ventricle, and then be pumped out to the body to deliver the oxygen and nutrients that's already present. There's also a secondary shunt, right, called the ductus arteriosus, which can shunt blood into the aorta from the pulmonary trunk. So instead of blood traveling through this pulmonary trunk and getting to the lungs, it can be shunted directly into the aorta and then delivered to the body that way as well. So the foramen ovale and the ductus arteriosus allows us to bypass that pulmonary circuit, to bypass the lungs, because that blood is already oxygen and nutrient rich thanks to the mother's body. So fetal growth is measured by measuring the fetus's crown to rump length, or CRL. So this is measured from the crown of the head to the curve of the buttocks, and a full-term fetus will have an average crown to rump length of about 14 inches. But they're going to be measuring this throughout the pregnancy just to make sure that the fetus is developing as it should be. And we know that the fetus will gain about 50% of its birth weight total in the last 10 weeks of development. So that's when it will actually start to pack on pounds and start to gain more adipose tissue or fat tissue for insulation right before it's born.